Hello everyone and welcome back to my channel. Today we are going to talk about SARS-CoV-2, the virus responsible for the ongoing COVID-19 pandemic. COVID-19 is a highly infectious respiratory illness that has caused significant global morbidity and mortality. In this video, we are going to discuss molecular diagnosis of SARS-CoV-2, which is an essential tool in the management and control of this pandemic. Understanding SARS-CoV-2. Before we dive into molecular diagnosis, let us first understand what SARS-CoV-2 is. SARS-CoV-2 is a highly infectious virus that belongs to the family of coronaviruses. It is transmitted through respiratory droplets and close contact with infected individuals. The virus primarily targets the respiratory system and can cause severe respiratory illness, including pneumonia, acute respiratory distress syndrome, and in severe cases death. Molecular diagnosis of SARS-CoV-2. Now let's talk about molecular diagnosis. Molecular diagnosis is a laboratory technique used to identify the presence of genetic material of the virus in patient samples. This technique is based on the detection of viral RNA in patient samples such as nasal and throat swabs, saliva, and septum. There are two primary methods for molecular diagnosis of SARS-CoV-2. Number 1. Reverse transcription polymerase chain reaction RT-PCR. RT-PCR is the gold standard technique for SARS-CoV-2 molecular diagnosis. This technique involves converting viral RNA into complementary DNA cDNA using an enzyme called reverse transcriptase. Simply, the cDNA is then amplified using PCR, a process that creates multiple copies of the DNA sequence. In PCR, fluorescent probes are added to the reaction mixture, which bind to the amplified DNA sequence and emit light. The amount of light emitted is proportional to the amount of viral RNA present in the patient sample, allowing for the quantification of viral load. RT-PCR is highly sensitive and specific, and it can detect even low levels of viral RNA in patient samples. Another detection method is loop-mediated isothermal amplification (LAMP). LAMP is a newer technique that has gained popularity for SARS-CoV-2 molecular diagnosis. Instead, LAMP uses a single temperature reaction, which makes it faster and simpler than PCR. LAMP also uses fluorescent dyes to detect amplified DNA sequences similar to RT-PCR. In conclusion, molecular diagnosis of SARS-CoV-2 is an essential tool in the management and control of the COVID-19 pandemic. RT-PCR is the gold standard technique for SARS-CoV-2 molecular diagnosis, while LAMP is a newer technique that has gained popularity due to its simplicity and speed. Both techniques are highly sensitive and specific and allow for the quantification of viral load in patient samples. Thank you for watching, and do not forget to subscribe to my channel for more informative videos on science and technology.